Yes, I enjoy getting my hands dirty because that's the only way you can get the job done. Let me tell you something about farmers in Africa. During the harvesting season, that is actually a happy season, a happy period. Farmers are very happy in this season because they are looking at the yield. It's a source of income for them. So they are very happy. So I have to be happy because it's something that generates food. It generates income for the farmer in its local environment. So this is a very happy period for me because I generate this data myself. I can be very sure of what I'm getting, not relying on what somebody else has generated. So I'm pretty much happy about these.
So instead of generating food, you're generating data?
I'm generating both, food and data.
Yeah, so we're in the tropical glasshouses at Sutton Bonington, future crop, and it's now 5 months since we last talked about the early stages of the crop. And now we're really at the harvest stage, so we're very much, most of the plants have been harvested, and we're starting to look at the individual plants to try and select the ones that we want to take on to the next generation. It's done by hand because and in Africa it would largely be done by hand, not entirely, but largely. And what we're doing is we're actually recording quite a number of the traits of the plants themselves, the individual plants. So we're looking at things like the number of pods, the number of seed, whether it's a spreading type or whether it's a bunch type. And all of that information will give us an idea of what genes are controlling some of these traits. So we can start selecting for the genes that are actually likely to give us the traits that are most useful in the field. Well, the first stage is really to make sure digging individual plants out which are numbered and labeled, and to make sure all of the pods actually come with the plant, because the plant has quite a long taproot. It can put pods under the soil. It doesn't have to, but it can. So we have to make sure we capture all of those pods. The next stage is really to take the pods off, to take the plant itself and dry it down, weigh it, work out how much biomass is there, how much light has been captured and converted, and then also the same thing with the pods. So how much of that captured radiation has actually gone into making something that we could eat.
You won't get to eat these, will you?
It's prepared as snacks, so certainly these ones are my research material. They're very precious to me, so I won't go to the point of eating these ones, but to do more research on them, to improve on these ones, so we can contribute to the local economy of the poor resource-based farmers in Africa.
Well, we've got some very interesting effects that are coming through when we change the length of the day for the plants. So the plants are generally speaking grown largely in tropical regions, and they have 12-hour days. But if you change that, you start to get effects in terms of flowering when it happens, but also whether pods set for particular varieties. And we are seeing very clear differences between different land races that have been sourced from different countries in terms of how they set pods, when they set pods, and what sort of yields they produce. One of the things it can lead to is it can lead to us trying to find material which doesn't respond as strongly to the effect of day length. And for most crops, most major crops, photoperiod is really one of the critical issues in terms of yield. And for this crop in particular, because if the photoperiod is wrong, it either delays setting pods, or in fact it doesn't set pods at all.
Well, there is time for everything. We actually know the time period for Bambara grows and matures between 4 to 5 months. Okay, so having been aware of that, there's no need to be in a hurry. It's a matter of time, letting the plants acclimatize to the environment, see if the plants are happy themselves. So when I come here during the growing season and I see the plants doing very well, it means the plants are happy. And in that case, I am very happy as well. So it's a matter of time. I just have to wait, be patient till the harvesting season for me to do the harvest.
Bambara in particular is quite drought tolerant, so it will grow where peanut doesn't. And in addition to that, understanding some of the genetics behind these things potentially allows us to improve yields. And for Bambara especially, that's yields for small farmers generally.
We're looking at a typical land race called Ankpa 4 that is found or originally came from Nigeria, a place called Ankpa in Benue State. This land race is very interesting because it's highly sensitive to photoperiod, meaning at 12 hours day length, it produces a huge amount of pods, but at 14 hours or 16 hours, it produces close to no pod. zero pods.
Well, we have controlled blinds, so we can actually control within the glass houses, we can control the photoperiod. So we've actually had this glass house here is at 12 hours, the one next to it was at 14 hours, the one at the end was at 16 hours. So we grow the same material in the different photoperiods, and we look at the differential responses.
So if we can get materials that are highly sensitive and ones that are very, very insensitive, we can carry out genetic crosses, develop populations that will segregate for these very traits that are of interest, that is of interest to us, and be able to identify the genes that are responsible for photoperiod. Could it be 1, 2, or 3 genes? We don't know. This is clearly not understood in Bambara, and that's why we are carrying out this research.